Welcome, everyone. Welcome to the latest edition of Fisher's Fishes. Today we'll be having a special lecture. Today's lecture is being delivered by Neeraja Sankaran, who is a historian of biology at Ahmedabad University. I happened to cross paths with her while doing like a short project with Jitesh Jhavar, also at Ahmedabad University. We literally just met in like the staff room or something. And today's conversation will be led by Swati, and Swati and Neerja are going to talk about Neerja's journey from the lab to the archive, yeah, where we trace Neerja's path from being mostly a microbiologist in the lab to someone who looks at biology with a longer view, let's say, looking at it from a historian's perspective and trying to unravel the mysteries of viruses and their parallel stories that we can tell. So I'll let Swati take over from here.
Yeah, hi everyone. I'm Swati. I'm Arhan's friend. But I'm also a PhD student in ecology and evolutionary biology at Yale. So when Arhan reached out to me about this, I was really excited because uh, even though I am not a historian of science, I happen to cross paths with a lot of historians of science, and so I really wanted to I'm just really excited to talk with Neeraja about her journey from scientist to historian of science and what that has meant for her academic career. So I guess Neeraja will kind of start. I think Arhan kind of introduced your background history. But for folks who aren't like as familiar, so Neeraja started her journey as a microbiologist at Punjab University and the University of Alberta. And then she did her PhD in the history of science and medicine at Yale, which is where I currently am. Uh, so I guess could you share with us a bit about the journey that you took? Like what inspired this transition from scientist to historian of science?
Sure. First of all, let me thank you for inviting me. It's a great pleasure to always talk shop um, in a time when I don't often get the opportunity. So there's that. And also, just it's always fun to spread the word. Yeah. So actually, the and then between microbiology and history of science covers a lot of years and ground. It
Mhm.
was not an immediate transition, but I went into microbiology as a undergrad. Growing up in India, you did science. You Science was one of the things one did. It was interesting. I didn't want to do math. My father was a math professor, so I didn't want to go into that side of science. I wasn't very good at physics, and biology interested me. And so I did a bachelor's and a master's. And for master's, I was in Alberta, as you mentioned, and it was a research-based master's. Uh, so based on writing a thesis, which a lot of you also, I think, these days, masters in sciences aren't just do a bunch of courses and get a master's. Neither was it in my case. But as I was doing the research, I could have maybe even taken that project on to a PhD. It was vaguely suggested, but that project had finite limits. You couldn't really do much more with it. The thinking behind it was done within the first 2 years. It was more about bacterial taxonomy, very, very uh, basic as in fundamental bacteriology. And it was interesting, but I thought, oh, maybe I wanted to do a PhD in something more humanly applicable, something more medically applicable. But one thing I very quickly learned in the lab is that the lab and I weren't necessarily best friends. I'm a klutz, and I don't do very well with repetition. Um, so I was an expensive person, I think, to have in the lab. I dropped a lot of broke a lot of glassware and dropped a lot of expensive chemicals down the sink. Um, but sometime during the masters, I figured out that what interested me about the science was the ideas behind it. and the doing of it was not necessarily my forte. Nevertheless, I persevered for a few years, tried to get to a PhD in a wider topic with a slight detour. Didn't quite work out. I mean, I got admission, but I wasn't, again, I worked as a tech while I was working towards admission and figured out again that the ideas were more interesting than the actual work. Pineapple leaves, I had that was in a plant pathology lab, and I had to grind pineapple leaves. And while eating pineapple can be a lot of fun, grinding up they're really tough critters to grind up, let me tell you. But in any event, I was working for a while in one lab, and then I thought, okay, before I did a PhD, maybe I should do some industry research. So I moved to the Bay Area and started to work in industry, and confirmed that I wasn't really cut out to be in the lab. But while over there, I found out about the science writing program, and that is actually was my first step away from lab science, but it was an important step towards eventually becoming a historian. History hadn't come into my radar yet. When I learned about the science writing program, this was a program, it was a grad certificate program at the University of California, Santa Cruz. I learned about it through an advertisement when I walked into a job center or something in the Bay Area Bioscience Center or some such thing it was called. And then I just saw this program and it was kind of the world stopped. That's what I wanted to do. And I abandoned all my job searches and talked to the director of the program who'd founded it. He gave me a lot of reasons for thinking that, hey, it would be very cool to join this. Then he turned it around on me. He said, okay, you've demonstrated your interest and I can tell why it's cool, but what have you? Yes, we need science. So this was a science writing program that mandated that the people getting into it needed to have a science background. And this sort of began this thing of answering questions that people put to me that are important in life without much thought. And sometimes that gut instinct serves to, and the reason, serves you well in the long run. And the reason I'm bringing this up now, because it served me again to get into history of science some years later. So I got the science writing program. He asked me, so yes, we want people with science, but we don't want them to come into this just because they don't want to do science anymore. You should have a better reason. And most people have worked with school newspapers, college newspapers. They've done some amount of formal writing. Have you? And I hadn't. So he said, what kind of public-facing writing do you do? And I thought on my feet very fast and said, I write about what I do to my grandfather. This was back in the days when one wrote letters, actually. Email had not yet been invented, or it had just started to be invented. And I don't know, maybe he thought I was being over smart, but he gave me admission into the program. And I had a really, really wonderful time for 1 year. We were dropped in the deep end and sort of had to scramble and learn writing. I learned some incredibly important lessons about writing and about myself through the program. The most important writing one being SDT, or show don't tell, which all legions of students, generations of students since then have seen this in their margins in the papers that I mark. But it taught me to really get an appreciation for telling a story, the program, how to tell a story and approach science from the perspective of a story. Fast forward a few years, and it was there during the program that I learned that there's this field called history of science and medicine. A colleague of mine, and a classmate of mine, I should say, was somebody who had started but abandoned a PhD in history of medicine. And I go, interesting that there is such a subject. But beyond the science writing program, I went for a couple of years and did an internship and all of that. And at some point, while working as a writer, figured out that while writing was interesting to me, I didn't get my dream job, though that came full circle or what I thought was my dream job. When I went into the program, I was like, great, I'll get this degree, I'll go and I'll start working for National Geographic and get to travel the world. We all have our dreams. That one I was disabused of very quickly because my professor told me people who get hired by National Geographic write captions for photos, they don't get sent around the world. Those are the naturalists themselves and the photographers who do so on their own dime. The magazine doesn't pay you to go around the world. And then that didn't interest me anymore. But this came full circle, if I may say so, very recently, when somebody from National Geographic interviewed me as a historian of science, and I've been quoted in the article on DNA. I'm the first and the last quote in the article. So I did get into National Geographic, albeit in a very circuitous and long after I thought I would. But learning about history of science and writing, and I loved writing, and it's very much part of how I see myself, I identify myself as a scholar. But at some point, I figured the stories one wrote as a journalist were short. I worked for a newspaper. I had to, finish. The turnaround was quick. I couldn't live with a story. And some of these stories I felt really needed living with. And then I started looking around for and then, I was brought up as the daughter of an academic, and I loved a lot of aspects of academic life, including the idea that you had long periods of time off, though that's no longer true. So I decided I did want to do a PhD, and I liked that life. See, the other thing is, when I became a writer, I thought, oh, I'll get to travel and stuff. But no, you did everything from your desk. You phoned people, you talked to them. I talked to a lot of famous people, including, for a biologist, it's exciting to be able to talk to Francis Crick and to be told that Francis Crick called me, which is actually just the way he did it. This was on the occasion of the death of Linus Pauling, for whom we were writing an appreciation. And Crick was one of the people we called for a report. I also got to talk to Jonas Salk, and to Carl Sagan. And he never said billions even once, much to my sorrow. But he'd gotten an award. So these were some of the very famous people I got to talk to while being a science journalist. But sometime during that time, I figured out that maybe I wanted to start looking for doing a PhD. But nothing came of it until 2 jobs later. I started with a newspaper, worked for a university information office, and while interviewing for a job at Rockefeller, having just lost another job, this person interviewing me, who was not in the public information office, but was also part of the interview process, asked me out of the blue, but if you didn't have to worry about money, what would you do if this job weren't a necessity? And I don't know why he asked me this question, but in the spirit of things, I entered it and I said, well, maybe I'd like to do a PhD, not even maybe, I'd like to do a PhD in history of science. And the reaction was, really? Why don't you forget about this job that you applied for? Work for me on soft money for a few years. You'll work on a historical project. You'll get a taste for it. And I can write you a reference letter and you can get into a PhD. And so 1.5 years later, that's what happened. I wound up at Yale. It was the second cycle of application. But that 1.5 years as what I call now a proto-historian was very important because, yes, it got me into the transition from being a straight writer of short stories to talking to people and collecting wider amounts of information, doing, even though we didn't call it that, oral histories, interviews. It gave me a lot of training. It also gave me in-depth exposure to certain stories in science that I didn't work on then, but that would surface later. And in fact, years later, became one half of my book, a copy of which I don't have to kind of wave to you. But the book is called A Tale of Two Viruses, and that was only published about 5 years ago. But so I went into Yale and I did a PhD. And doing a PhD at Yale, as Swati will know, is wonderful because those first 2 years, you get to take whatever courses you want in whatever department you want. I needed to catch up on history because history of science was not something history was something I hadn't studied since grade 10. So I took a few history methodology courses and the core history of science classes, broke my head over Michel Foucault, tried to read, we didn't do that much STS. So Bruno Latour hadn't yet become, for me, that big a thing. Foucault, however, Birth of a Clinic, I certainly cut my teeth on that. Did a vast amount of reading in history of molecular biology, medicine, because it was Yale and Yale's history of medicine program's really great. This advisor that I began with, Larry Holmes, was an absolutely wonderful first mentor to have in history of science. A very renowned historian of science who's done these mega tomes. And he gave me a lot of very good advice as a historian, including, while complimenting me, pulling back on the compliment on my ability as a science writer, but he told me to, really also get my credentials as a historian. And so it's thanks to him, I think a lot of that, my training as a historian. But then he unfortunately fell very ill and passed away. And even before he died, another wonderful scientist who's a historian primarily by affection, by name of Bill Summers, who had written a biography of the bacteriophage, discoverer of the bacteriophage. He became my primary advisor. And, but even before that, before he became my advisor, my project had been set because of something a Nobel laureate told another historian who told a bunch of us grad students at coffee. So always as grad students, go to coffee with visiting speakers, because you get a lot of good ideas. I got my thesis topic because she was sitting there and saying, Joshua Lederberg says that the work this immunologist McFarland Burnett did on bacteriophages, bacterial viruses, is really understudied. Nobody knows about it, and it's worth a great thesis. And I glommed onto that idea and I talked it over with my advisor. He said, okay, go do some preliminary research on that. But he also gave me 2 other projects. And he said, do some projects on that as well. I'm talking a lot, so if you need to cut me short and interrupt, please do so.
Absolutely no.
So
We
he
don't
told
need to.
me to also look at a couple of other projects. He gave me, another person I was kind of fascinated by was JBS Haldane, who has lived in India, was a geneticist, but a biochemist before that who had done work on origins of life. And as I found out only at that point, was the author of a very favorite book I'd read in my childhood, a piece of fiction called My Friend Mr. Leakey. And he has the best description of eating. And now that it's mango season, he talks about Mr. Leakey, the magician, introducing himself, the protagonist, to mangoes and how the best place to eat them was sitting in your bathtub. And so I wanted to do something about Haldane, but Larry Holmes, that was my first advisor, very wisely pointed out that Haldane is a lifetime project you'll never get done, because he's done so much that you'll have trouble focusing on what aspect of his work to focus on this thing. But he said Haldane had a very interesting father who did very interesting work in physiology. So why don't you check that out? I did. By the way, Haldane and his physiologist father are characters in a Aldous Huxley book called Point Counterpoint. I think it's called Point. One of those satirical books about academia. It's a wonderful piece of fiction. So, where the character of the father will come in the middle of the night waving some frog or something, whatever his experiment is doing in the basement. So Haldane was another possibility. But I did some preliminary research and ultimately went back to Burnett. I was interested in immunology at that time, and bacteria and molecular biology. And Haldane's work was much more respiratory physiology. And then there were one or two others, and it'll show you that I can't even remember those people's names anymore. When I went back and told him, by then he was quite ill, but still still the head of my PhD. He hadn't handed over the reins yet. Bill Summers, who eventually became my advisor, was on my committee by then. But I went and talked to Larry about this because he was still coming into the university. And I said, I showed him what I'd amassed, and I said, yeah, this is the one I want to do. And he started laughing gently, saying, yeah, I kind of thought that's where you'd end up. It's a really good project, but I did want to push you to at least consider something else and not develop tunnel vision. And one of the good things about that project was that it was naturally bookended. There was a big, he had a first paper, and there was a time when he just stopped doing bacteriophage work. And so that became my thesis over the next few years. I got a trip to Australia because of that, because Burnett had worked in Australia all his life, and Yale has only the second largest library in the world, the largest academic library, the largest, I think, being in Harvard. But of course, I found an archive clear across on the other side of the world to go and do some work in. Did that, enjoyed that thoroughly. So that was, I think, my first real work in an archive, digging through papers, making notes about what I would want later, regretting that I hadn't, later in life regretting, I thought I saw that, but where it was, and how can I go back and dig, learned the value of digitizing. This is all well before AI. He had this I read copious amounts of letters, numbers of letters, personal letters, some of which read like they could have been written yesterday, and others of which are very dated. He kept diaries. I got to read those. So, I've suddenly found out about learning about science from many different ways, not just lab notebooks, which was Larry Holmes's forte, but also this young man as a medical student writing whatever he's looking at. He's quite a nerdy person, Macfarlane Burnett was, because one of his letters to his then fiancée, later wife, was, this is my 137th or some number, treatise on bacteriophage, your chief rival for my affections. So he was quite the nerd. But reading letters and finding out about how people think about science, it really made him, and it gave me a whole different window into science writing, because he was a very good writer for public as well. And, in writing to his fiancée, who's a nurse, about this very esoteric, this is in the 1930s, late 20s, 30s. Bacteriophage was only found in 1915 or 17, depending on whom you ask. Viruses that infect bacteria. And by 25, they'd become a plot device in a piece of fiction. But other than that, very few people knew about it. He himself, even though he was very young, in his 20s and early 30s, in his 20s at that time, was able, was demonstrating this to all sorts of dignitaries, that what this thing could do. How could you see it in the lab? Because you can't see viruses. How do you see its effects? He did some demonstrations. He studied it. He went to England, got a PhD in it, came back to Australia, did some more work with it, but then got sidetracked. There was this incident where there was some contamination of vaccines, and you can imagine what kind of field day anti-vaxxers would have had with it. But he was able to very quickly resolve that issue, and that took him off onto a slightly different tangent, studying bacteria that could cause contamination, began to get more deeply into bacteria, also got into viruses and became a very famous, actually, virologist, studied influenza virus. Anyway, so he did all that. But once he stopped bacteriophage, he stopped it for good. And there ended where I needed to end my thesis. And so I wrote my thesis, took me a good many years, learned a lot about other issues on the way, history, biography, got really interested in, took a class on early modern autobiography and learned a lot about, how different people wrote, again, letters are, and used that over the years to create courses on biography, autobiography, how to put them together to be a historian. Learned all this. So Burnett's papers. And then while I was working on that, some things about the way he was thinking about bacteriophage and the way bacteriophages were understood made me think back to, this has some parallels with the way something I'd worked with during my proto-historian days on cancer viruses, which have nothing to do with bacteriophages. And so many years after I got my PhD and then I did postdocs, I moved all over the world. Actually, straight out of my PhD, my job was not in history of science. It was to teach writing, science writing. And being a writer and teaching writing are two very, very, very different things. And you, just because you're good at one does not mean you are automatically good at the other. Writing, I became fairly decent at, I was a competent writer. I was a good writer fairly quickly. Becoming a good writing teacher took a lot longer. And it wasn't always something I wanted to do, but I did it because I think it's very important to teach, no matter what you're doing. I studied writing to be a writer, but even if you're not going to be a writer, I think studying writing is very important. And even in this day and age, when you can normally feed AI the prompts and get it to do it for you, there's still some value in being able to do it well. And also to be able to give it good prompts, you need to write well. So, so I did that. And so I'm fast forwarding many years after I got my PhD, I thought about these parallels, floated it at a meeting where a friend, and called it A Tale of Two Viruses, and very diffidently said, it was the best of times, it was the worst of times. And a friend said, Neeraja, if you're going to use that parallel, own it. Don't be so diffident about it, because, it isn't something to be diffident about. And sure enough, once I took ownership of that parallel, I found even crazier parallels, including bacteriophage as bacteriophage as Madame Defarge, who's one of the most fearsome villainesses of all time, but who's a villainess in the book A Tale of Two Cities. So there's even a cartoon in my book showing, so the reason there's such a great parallel is Madame Defarge used to sit at every guillotine session and be knitting. And what she would be doing is knitting a register of all the intended victims of the guillotine. And the bacteriophage knits itself into the bacteria and into the DNA of the bacteria, knits its own DNA, and thereby earmarks it for the guillotine. So I thought this was a very cool metaphor, and I ran it by a French teacher, a French scholarship, a French scholar, Michel Morange, and he says, that's great. Definitely bring that into your book. So eventually I did, and then a wonderful cartoonist friend of mine, who's a microbiologist, drew me the cartoon. So that became part of my book. So I've kind of wandered all over the place. So I became a historian largely of bacteriophage to begin with, but that was not a book in and of itself. I broadened it to be of viruses, and therefore became a historian of virology, but also of molecular biology, because that was anyway the kind of biology I had been doing. Sometime along the way, I realized people aren't reading papers original papers very well. And that became a big thing for me. And I wanted to actually teach a course. I started to teach classes, which I called texts in context, where it didn't matter what level so reading original text was something that we were taught to do at Yale. We did do classes with that. But this idea that students, even at the undergrad level, should not just read a text, but also read the way so that they learned about my field, history. Look at how a historian is thinking about that text, how somebody who's not a scientist primarily has written about that discovery. And so starting to juxtapose these things made me realize that this is a really good way to bring the really interdisciplinary way to bring these subjects to scientists, where they can learn about the history of the field, but also to teach, get non-scientists to read original scientific papers, and then they can read about what is being thought about them. And at some point, I wanted to do this for DNA because one of the big projects I worked on. So while I was at Rockefeller doing that proto-historian work, there were two big projects that I worked on. And the way the proto-historian work there, going back in time, was to celebrate in preparation for an upcoming centenary of the university. So I worked there in 95, 96, and the centenary was going to be in 2000. So I did a lot of base work. And one of the things I did was to start to put together dossiers or, material on various famous scientists who'd worked over there. And one of them was Oswald Avery. That's the project I began with, who, together with his co-authors Colin MacLeod and Mac McCarthy, had published this paper in 1944 that shows that DNA is the material that is the stuff of genes, to borrow their words. Now, everybody associate DNA with Watson and Crick, but not all they did something really important, but what they did was find the structure of DNA. But why they were even looking at DNA, because it was not considered an important molecule for, if you think about it, they found the structure of DNA in the 50s, 1950s. DNA as a molecule was discovered in the 1870s. So 1869 to 70 is when this substance called nuclein was discovered by this guy called Miescher. And the National Geographic article I was telling you about was, in fact, about Miescher. And I was quoted on his contribution. So DNA had a really long history. And I felt, again, people just begin to talk about DNA with Watson and Crick. Avery was tremendously important. In fact, his paper has been deemed as the most important paper for biology in the 20th century. And it occurred to me that maybe I should do this text and context for the whole story of DNA. And beginning with Miescher. And when I went to look for Miescher's paper, I found out that it was in German with the very nondescript title of, I'm translating it because I don't have the German in front of me, on the chemical composition of pus cells. With a title like that, nobody, unless they are intimately already aware of the history of DNA, would think to go look at a paper with that title in a history of DNA, right? And yet it's the paper that shows that announces or shows the discovery of the chemical that turned out to be DNA and how to purify it from cells. 1860s. And then I talked to another friend who also lamented not knowing, a couple of friends, one of whom was half German, and he and I therefore translated this paper and wrote a commentary for it. While I was thinking. And then this idea grew to maybe make this course. I thought about it. It didn't quite fly as a course then. It has since been taught as a course. And then I was giving a lecture on a totally different topic, the title of which you might enjoy. It's called Fantastic Microbes and Where to Find Them. And I can talk a little bit about that when you ask me questions. I think I should wind this up. But in any case, so I thought about this DNA very, and I thought maybe it could be, this guy was there who really liked my talk and came to talk to me, and he was an editor for Audible. So I told him, why don't we, at that point, it wasn't with DNA, it was with those papers that I'd been discussing. And I said it would be really cool to have somebody read these papers and then have discussions of these papers. And at some point, it morphed from being an idea for an audio book into an idea for a podcast series. And so this idea of the DNA papers podcast was born kind of as a podcast modeled along the radio show In Our Time by Melvin Bragg. So my idea was I'd be Melvin Bragg, haha, and invite a bunch of people, a scientist, a historian, a public-facing individual. So 3 to 4 people to talk about a specific paper or cluster of papers in DNA. And so I built a narrative arc from beginning till about discovery, a little beyond the Watson and Crick structure. So Watson and Crick actually are episode 13 in a 15 episode podcast, just to give you some sense of where. And episode 7, central episode, belongs to Avery. So there were 7 papers leading up to it, 7 beyond that. So for the purposes of the podcast, it became conversations around the scientific papers. In a classroom, it's translated again. Each of those papers is paired with a history paper. And I've done this with other subjects in class. I've done it with microbiology. I'd love to do it for viruses. And it's a good way to get students to think about their subject in multiple contexts. So I've become a historian. I worked in archives, dug through, looked at papers. Burnett's not the only one. Peyton Rouse was another one, the guy who discovered the cancer virus. That is the other half of my tale of 2 viruses. I've gone all over the place. I think I better let you ask me questions to bring it back.
I mean, no, thank you so much. I feel like you've covered a huge ground of like, so some of the questions I wanted to ask you later on were like specifics about the book, but you've covered so much, and I think this will be really, this has been really informative for all of us. Something that I wanted to ask you. So I was struck by what you said about how you were interested in the ideas behind the science. And maybe this might be an artificial distinction that we make when we think about the process of, or like the history of the ideas behind the science versus like the development of the actual ideas. So I guess I just wanted to ask you, like, at what point did you realize that, like, did you make that distinction of like you were actually interested in the historical process of how these ideas were developed versus like wanting to actually develop the ideas of like interested in the developing these scientific ideas?
No. So that to me is not that distinct. For me, it
Okay.
was the distinction of the ideas versus the getting hands dirty. For me,
Okay.
the history versus the present development is not a big distinction, because I think history happens as we are
Right.
doing it. So the idea, anything, it's like evolution, right? Every
Right.
species is a transitional species between what came before and what's going to come next. There is no such thing as a non-transitional species.
Right.
And it's similar history. I mean, an idea is always developing. And it's never quite set in stone. At least that's what we like to believe. But the way we think about viruses is still happening, and I'm interested in how it's unfolding even now. But yeah, I can't really understand that until I know what happened before. That was the kind of, that's where the distinction, like if I start to write a book and a history, and I want to cover period this to that, I always have to go back a few decades or a few centuries before the so-called starting point. Now, how I did it in my book was to start with the discovery, but then by chapter 3, kind of give a backdrop to the context of that discovery, what was already being thought.
Yeah, no, that makes sense. I guess I wanted to ask that question because I think as scientists today, very few of us actually, go into like understanding the history behind those, or like how those ideas developed. And like the way it seems to me nowadays, at least, is that, to understand the history behind these ideas, you become a historian of science. And like that separation of disciplines is how we think of it these days, even though it probably shouldn't be, because I think
I
something that,
agree that it shouldn't
yeah,
be, but
yeah,
these days is also a little. I mean, history of science wasn't even a history of
mhm,
medicine has a long and venerated history,
right,
but history of science really began with two people, Thomas Kuhn, who did it for physics, right? The Copernican revolution, and wrote that book, Structure of Scientific Revolutions, and Sigerist, who set up the History of Science Society and started to think about it historically. I think what you're just saying is true. Everybody needs to, but not very. There are very few scientists. Burnett, the guy on whom I had the good fortune to do my PhD, was one who actually thought actively about the history and philosophy of the topic.
mhm,
Many others don't. You know, or they rewrite history. I mean, one of the most famous so-called history books in molecular biology is a book called Phage and the Origins of Molecular Biology, which was a collection of essays put together in honor of Max Delbrück's 60th birthday. The point is that it's a great book, but it's all reminiscences by scientists, and it lacks the historical rigor that a historian would expect. I mean, it lacks the kind of rigor of analysis, historical analysis, right? I mean, and it always is told in the like history is written by the winners. They say in other fields, and certainly in phage history, that sort of became the truth for a long while until historians decided to provide the corrective and show other stories. And so I think, yeah, it is important which is why I've been trying to push these kind of courses, right? You read the original papers, you read the historical papers. This is a class that senior PhD, senior master students and PhD students, wherever I've taught, PhD students at NCBS, a few PhD students here at Ahmedabad University, and senior students. These are the students who are taking those, or who have taken. Not a huge number, but even small sparks hopefully will start the spread.
Yeah, and that's sort of one side of, things we should be doing but don't do. Sort of to me, the other side is also, and I think we've spoken about this before, is communicating the science we do as like, not as we do it necessarily, there'll be some lag, but still it seems to me like most scientists, not only do they not communicate, if you go and read papers now, sometimes it feels like they're written to not be comprehensible. They're written to be like
Oh,
defending
very
against
boring.
some.
Um,
Yeah.
yeah. So, I
I
mean, that's something I could talk about. I have a lot of friends who could talk about that even more than me. I mean, one of the cases in point is this paper whose title I mentioned, right? On the chemical
Yeah.
composition of pus. It has some great ideas thrown in at the end, but you'd never know to look for them. Because, well, first of all, not his fault. It was a chemistry paper, and it goes painstakingly through the procedures of what is being extracted, how much ash there is, how much potassium, how much, not potassium, carbon, hydrogen, phosphorus, blah, blah, blah. So, and how he's ultimately comes to the point about why he thinks this chemical is unique and why it's important. Those are 3 or 4 lines in a 28-page paper.
Mm-hmm.
And that was, I mean, this was a report, but somebody, even himself, should have been writing about it. I mean, he wrote a lot of letters, so you can see it sort of distilled or made easy. But I think science communication is incredibly important. And in fact, I often think of, if I, make up courses in my head, one of the courses I think that I would love for every science student to do, an exercise anyway, is to, I've tried to set it for some people, is to think about different audiences for whom they're writing the same thing. You're doing experiments. How do you explain it to a lab mate? How do you explain it to somebody? How do you justify it to somebody who knows much more about it than you do? To a professor who's not in your lab, but you justify it. So that's writing for a audience that knows more than you do. And a way to think about that, instead of just justification, a real world way to think about that is, okay, your thesis is a contender for the prize. Write this professor a short note justifying why you think your thesis should have a prize. So you immediately start to think of reasons about why the work you're doing is important. And then, of course, there's the largest stakeholder is the general public. So you have your peers, your superiors, and the public. There are at least 3 audiences you should be writing to. Where and how, I mean, these days, we have so many different ways. A forum like this is actually a good one to maybe even, I mean, you're running a series talking to people like me, you should be running a series decoding work that all of you are doing. A paper that you're about to publish, or some work that's being done in the lab even before publishing or post-publication. What makes it important? One of the first assignments I had as a science writer was an editor at The Scientist. They had a section called Hot Papers, and I was made editor of that section. Hot Papers used to take out, look at statistics, because it was newspaper was founded by a guy who started the Institute for Scientific Information that puts out current contents and looks at citations. And papers that were deemed hot in a topic that had received a lot of unusually high number of citations compared to other papers in that field within a very short period. And usually they asked scientists to write about why they thought it was important, and the things were deathly dull. But once we got our hands on it, I started to talk to the scientists, why do you think it's important, and rewrite it that way. And it immediately became a lot more accessible. I was still called the editor. We used to have wars over, not wars. The owner of the paper wanted to call me the editor of the section and still give authorship to the scientist. A few other people didn't think that was fair, but the owner's gone. I no longer work there. The paper's moved. So it doesn't really matter. The point is, Hot Papers became a more meaningful section once you were able to provide that liaison. And I think every scientist owes it to themselves, really. And one way to do it, I think, is to try and talk to, and this is something I tell my students also, get somebody who isn't in the know, isn't in your lab, and explain it to them verbally first and then write it down. Or write it down and ask them to throw it back at you. What have they understood? And always write a short piece that I get kids to do in my writing classes is write about what it is and the so what. Write a short what, so what for everything that you're doing. So why is it important? Who is it important for? The so what will start to bring up a lot of different questions and aspects. So did I digress too much, Arhan?
No, actually, like what I was looking for was basically an understanding of what are the different ways we can think about doing it. Because ultimately, you do have a lot of people in this room who want to be scientists, and communication is a part of it. And if we
Absolutely.
have these ideas, as you said, if you have these ideas diffusing out slowly, you might see them spreading. So I just
There
wanted
is
to,
that, but there's also writing your own papers well.
yeah,
And I think all labs should have some sort of regular meetings about trading writing, brainstorming ideas for writing, but also maybe reading, because reading makes you a better writer. So choose a book
yeah,
that is not a textbook, but choose a good book, start to write it, start to write reviews. I mean, start to read for how are the things explained there, because writing well works best using models.
yeah,
So, pick an older paper in your field. I mean, you should actually pick two models, a model of what you want done and a model of what you don't want your paper to look like. Actually, a bad paper is a very good teaching device, learning device, if you would.